This one, these are type of pictures that like this certain motility without anybody. So like pictures of this. Like they're taking pictures of food. But then
Me, I did not eat too.
I'm not rushing. I'm not taking my laptop.
Who's that was?
I went back to
Not sure my. Not sure this one. We can show that I can always. So, the summit of the experiments, do the sperm somatozoa analysis. We have the motility checks for each group, and eventually we take blood samples, and the blood samples are kept in paralyzed tools, paralyzed tools, and kept for analysis, food analysis. Then we have the surgical instrument, the setting sets, which have been used to have the excisions done. So we have excisions, especially for this particular study is centered on excision for this study is particularly centered on epididymis and testis. That is the main one. Then we now take the blood sample that I did mention. So for each group also in this experiment, we have 6 groups, control inclusive. Then we test for different treatments. Especially we use ethylene glycol monoethyl ether. So that is the main treatment. Then we counter we use a therapeutic treatment. The therapeutic treatment is chlorogenic acid, and chlorogenic acid is a phytochemical that is obtainable from plants, most plant species. So we've been able to check the effect. So after this time out now, we'll now move on to other analysis where we do the morphological studies and what do you call it, the reproductive assays. So once we have the reproductive assay, we will now be able to move on to the histological aspect of, the statistical aspect of it. That is where we deal with the biostatistics and know how effective, truly, how significant the study has been. If these studies can be moved to the next level of the phytochemical, of pharmacological studies, where we now say, okay, we can now use human models instead of using animal models. So we have to use human models to ascertain the efficacy of the chlorogenic acid, which has been used as a therapeutic studies here. So still stay tuned while we move on. So like, share, and subscribe to our channel. Thank you.